The pharmaceutical industry is a critical component of modern healthcare, responsible for the development, production, and distribution of medications and other healthcare products. This industry plays a vital role in improving patient outcomes, managing chronic conditions, and saving lives. The pharmaceutical industry is responsible for the development of new treatments and medications, which can help manage or even cure a wide range of health conditions. These treatments can range from simple over-the-counter medications to complex drugs used to treat rare diseases. Chronic conditions, such as diabetes, heart disease, and cancer, are among the most significant healthcare challenges of our time. The pharmaceutical industry has developed medications that help manage these conditions and improve patient outcomes, allowing individuals to live longer, healthier lives. The pharmaceutical industry has played a vital role in preventing disease through the development of vaccines and other preventative medications. These treatments can help protect individuals from serious illnesses, such as polio, measles, and influenza. The pharmaceutical industry has also developed medications that can significantly improve quality of life for patients with chronic conditions, such as arthritis, multiple sclerosis, and depression. These treatments can help reduce pain, improve mobility, and enhance overall well-being. Differential scanning calorimetry is a thermal analysis technique that measures the amount of heat absorbed or released by a sample as it is heated or cooled. This information can be used to determine the melting point, crystallization temperature, and other thermal properties of the sample. In the pharmaceutical industry, DSC is commonly used to analyze the thermal behavior of drugs and excipients, and to identify potential stability issues. Differential scanning calorimetry works by measuring the difference in the heat flow between a sample and a reference material as they are subjected to a controlled temperature program. The sample and reference materials are placed in separate pans, which are then heated at a controlled rate. As the temperature of the sample and reference materials increase, the DSC instrument measures the amount of heat absorbed or released by each material. The difference in heat flow between the sample and reference materials is then recorded as a function of temperature. For drug development, DSC can be used to study the thermal behavior of drug molecules, including their melting points, glass transition temperatures, and enthalpy of fusion. This information is useful in drug development to determine the best formulation and processing conditions for a drug. For quality control, DSC is also used in the pharmaceutical industry for quality control purposes. It can be used to ensure that the physical and chemical properties of a drug remain consistent over time. For stability studies, DSC can be used to study the stability of drugs under various conditions, including temperature and humidity. This information is useful in determining the shelf life of a drug and in designing appropriate storage conditions. For polymorphism studies, DSC can be used to study polymorphs, which are different crystal forms of the same molecule. Polymorphism can have a significant impact on a drug's properties, including its solubility, bioavailability, and stability. Differential scanning calorimetry can help to identify and characterize different polymorphs, allowing researchers to select the most appropriate form for drug development. For compatibility studies, DSC can be used to study the compatibility of different components in a drug formulation. This is important because incompatible components can cause degradation or changes in the drug's properties. By using differential scanning calorimetry, researchers can identify any potential compatibility issues before the drug is developed further. For formulation development, DSC can be used to develop and optimize drug formulations. By measuring the thermal behavior of different components in a formulation, researchers can determine the best formulation for a particular drug. In the pharmaceutical industry, accurate DSC analysis is essential for several reasons. Firstly, it helps to determine the thermal stability of drug compounds and their compatibility with other excipients. Secondly, it assists in the development of drug formulations with optimal properties and stability. Finally, it aids in the characterization of polymorphs and the determination of their impact on drug efficacy and safety. Crucibles are an essential component of DSC experiments as they hold the sample during the heating or cooling process. Aluminium crucibles are commonly used in DSC experiments due to their high thermal conductivity, low cost, and ease of handling. However, aluminium crucibles also have some limitations that must be taken into account when designing and interpreting DSC experiments. Aluminium crucibles have become the preferred choice for DSC analysis in the pharmaceutical industry due to their superior properties over other materials. Aluminium is an excellent thermal conductor, which allows for rapid and accurate heat transfer between the sample and the sensor. Additionally, aluminium is highly resistant to corrosion and does not react with most chemicals, making it ideal for use with a wide range of drug compounds. Aluminium has a high thermal conductivity, which allows for efficient heat transfer between the sample and the sensor. This results in more accurate and reliable results. Aluminum is highly resistant to corrosion and does not react with most chemicals, making it ideal for use with a wide range of drug compounds. This ensures that the sample remains uncontaminated during analysis. Aluminum crucibles are significantly cheaper than platinum and ceramic crucibles, making them a cost-effective option for laboratories. Aluminium crucibles are easy to clean and maintain, reducing the risk of contamination and ensuring consistent results. They are available in high-purity forms, which eliminates the risk of contamination from impurities in the crucible. Differential scanning calorimetry is an important technique while the aluminum crucibles work as key consumable supplies, used in the pharmaceutical industry to ensure the quality and efficacy of drugs. By analyzing the thermal behavior of drugs and excipients, DSC can help to identify potential stability issues and optimize drug formulations, while aluminium crucibles provide accurate temperature control and prevent sample contamination. Ongoing research and development in the field is helping to overcome the challenges associated with DSC and improve its usefulness in pharmaceutical analysis. Thank you for watching. Feel free to follow our channel and visit our online store. If you have any questions or needs, we will promptly answer and meet them for you.